We will move on now to Dr. Ivano DiMeo. He will be speaking about a new mouse model for CoPAN. Take it away, Ivano.
Okay, thank you, Susan, and thanks to the organizers for this opportunity. So in this talk, I'm going to show you our latest data about the generation of a new, the first mouse model for CoPAN. So you will know this slide. I'll go very fast. Other than PCAN, counting the a big group of NBIA disorder, we found some years ago the first patients with mutations in CoAC gene encoding for the Coenzyme enzyme, catalyzing a bifunctional enzyme, catalyzing the last two steps of Coenzyme A biosynthetic pathway. So the two diseases are pretty similar, as mentioned before, but CoPAN seems to be extremely rare and more severe than PCAN. Probably, maybe because there are more pan-K isoforms, but only one CoAC gene. So we reasoned that the implementation, the generation of a mouse model lacking CoAC gene could be informative, interesting to study the pathogenesis of the diseases associated to inborn errors of Coenzyme A. So we generated a mouse model using the classical Kryllox system. And as expected, we found that the constitutive ablation of CoAC gene in all the body of the mouse is not compatible with life because it was expected because this is a very important gene. So we decided to use some tissue-specific transgenic mice expressing the Cre under the control of tissue-specific promoter. Here I show you some data about the use of a neuronal-specific Cre expressed under the control of the synapsin-1 promoter that is expressed in the neurons starting from the embryogenesis. So the mice showed a reduction, a deletion of CoAC gene in about half of the total brain. That is more or less the amount, the part composed of the mouse brain composed by neurons. And also the expression of the gene, also the protein, is more or less half, 50% compared to control littermates. So it is important to keep in mind for the rest of the presentation that only the neurons lack CoAC, but the other cellular lines, such as the glia, the astrocyte, the oligodendrocytes, etc., possess normal CoAC gene. So clinically, we found that syncoAC animals are indistinguishable from control littermates at birth, but they start to develop a very severe phenotype, clinical phenotype, very soon, at about 6-7 days after birth. This phenotype is characterized by a growth arrest, an alteration of the sensory motor behavior, and the appearance of movement disorders, as you can see in this video, that is characterized by the inability to correctly ambulate, some dystonia like movements, and also some rigidity or spasticity. And very important, a very short lifespan with a median survival of about only 13 days. So this is a good news because we generate a model of CoPAN with a clear clinical phenotype resembling the human phenotype, the patient's phenotype. But on the other side, this is also a bad news because it is very difficult to study, to work with a mouse model with a so strong, a so severe phenotype, and also especially with a so short lifespan, especially when we are talking about neurodegeneration. For example, indeed, we didn't find any sign of neurodegeneration in the brain of knockout animals, and also the completely lack of other signs of brain pathology such as astrocytosis or neuroinflammation. Probably this is due to the short lifespan of the mouse, so the brain doesn't have the time to develop a brain pathology, a neurodegeneration. So biochemically, we at first looked at the biosynthesis of the coenzyme A, and with our big surprise, we didn't find any alteration. We found normal levels of CoA and acetyl-CoA in the brain, in the forebrain of knockout animals, and also normal level of protein acetylation derived from the acetyl-CoA. Actually, at the moment, we don't have an explanation for these results. We are looking for an explanation. We are studying this aspect right now. For example, we analyzed the expression of the other gene involved in the CoA biosynthetic pathway, looking for a sort of compensation mechanism, but we didn't find any alteration of the expression of the other enzyme composing the pathway. So this is a very intriguing aspect that we are investigating right now. Next, we looked at the iron metabolism, because you will know that iron accumulation is the hallmark of the disease, and we found a global alteration of proteins that are involved in iron homeostasis. The most altered protein is the ferritin light chain. Ferritin, you know, that is the complex that store iron in the cytoplasm, in the cell, and the levels, the ferritin levels are associated to the levels of intracellular iron. We found that ferritin accumulates mainly in the neurons, mainly in the cortex of the knockout animals, but also in the subthalamic nuclei and in the midbrain and medulla, but very low accumulation, or sometimes no accumulation of ferritin was found in the striatum. And in the cortex, we found that the ferritin accumulates mainly in GABAergic neurons, often as these cytoplasmic granules. Other proteins that are altered in the brain of knockout animals are the transferrin receptor 1 and the DNT1 protein. Both are involved in the iron uptake, and we found a slight but significant reduction of both these proteins. And all the expression, the RNA of these proteins are normal, so as expected, this alteration is occurs post-transcriptional. But we, at the beginning, we were unable. So all these this alteration suggests iron alteration and maybe the iron accumulation, but we were unable at the beginning to observe, to analyze, to measure and observe evident accumulation of iron by typical approaches such as the Pearl staining or spectrophotometric analysis. So we performed, in collaboration with the group of Sophie Essiro in Lyon in France, we performed a secondary ion mass spectrometry imaging, so a mass spec imaging assay, and we found low but appreciable accumulation of iron through all the brain, and in the same area, you can see here, also an accumulation, more evident accumulation of calcium that we found also accumulated in patients of PECAN and CoPAN, and also magnesium in the same area. So next, by the same imaging, the same approach, we found also a dysregulation of lipids in the brain. For example, here I show you a strong, evident reduction of cholesterol in the white matter of the medulla of knockout animals. At the end, we are also analyzing the mitochondrial function, because you will know that coenzyme A is mainly used and stored in mitochondria, and also PANK2 and CoASY are also associated to mitochondria. We didn't find alteration of the single complexes activity of the respiratory chain, but we found a global alteration of cellular respiration by analyzing the OCR, the oxygen consumption, giving to the brain isolated mitochondria as substrate pyruvate and malate. So analyzing all the flux through the TCA cycle to the respiratory chain, and oxygen consumption in knockout mice is decreased. And also we found some altered mitochondria in morphology with this completely altered crest in the brain of knockout animals. So in conclusion, at the moment, we can say that the neuronal specific ablation of CoASY in mouse leads to the appearance of a severe, very severe clinical phenotype that is characterized by the appearance of movement disorders, sensory motor defects, and a very, very short lifespan, but without any signs of neurodegeneration or, in general, brain pathology. So we are working to now, in order to generate some different models using an inducible approach to trying to extend the life of knockout animals. Surprisingly, we found normal levels of CoA and acetyl-CoA in brain, and we are investigating right now this situation, but we don't have at the moment data. A dysregulation of iron homeostasis and an accumulation of iron, initial accumulation of iron and calcium and magnesium in brain, a downregulation of cholesterol, and an alteration also of mitochondrial respiration and morphology. And I want to thank all the people involved in this project, from our institute, our laboratory, but also the group of Sonia Levi from the San Raffaele Hospital in Milan, the group of Sophia Essiro for the SIMS imaging experiments, and the group of Nico Mitro for the mass spec analysis of coenzyme A and acetyl coenzyme A. And thank you all for the attention.
Thank you very much, Ivano. We are at time, but I'm going to ask just one combined question from Chuck Rock, who was the first one in with his questions. And then I'll ask you and the other speakers to please monitor the chat and the Q&A. There are questions, Ivan, Ivan, for you, and Ivano, I'm sure that there will be questions for you. So this is a combination question in the experiments where you measured CoA levels. How old were the mice, and do you know the cause of death in your mouse model?
No, we do not have, we don't know the cause of the death of mice. The phenotype is very strong, so we don't know if there is a difficulties for mice to eat or something else. We really don't know. And the second question was?
Yeah, so it was in the mice where you measured CoA levels, how old were the mice?
How we measure CoA? We measure CoA by different methods. At the beginning, we used an HPLC method and also the commercial.
What was the age of those animals?
At the end, 10, 13 days.
Okay, thank you very much.